All right. Let's move on to chapter 17. So we've talked a lot about innate immunity, and now we're going to talk about the other part, which is adaptive immunity. What is the most sort of central, important part of this kind of immunity, would you say? What is the thing that our body makes or does that is really key to allowing us to have this kind of adaptable immune system? Antibodies. Does anybody know what cells in your body make antibodies? It's a type of white blood cell. Yep. Any specific ideas about which type of white blood cell? It is a lymphocyte, and there are two kinds, B cells and T cells.
T cells.
It's not T cells, but that's a great guess. If you only had two, why not? It's the B cells. Those make the antibodies. That's how I remember it. But T cells help them to decide whether or not their antibodies are a good idea. Okay. So as I said, we've talked about chapter 16, which is innate immunity, and now we're moving into chapter 17. What's the major difference between these two artificially separated branches of our immune system? What's the main difference between innate and adaptive immunity in terms of how we're responding to things? Yeah, so the innate immune response is a whole bunch of tools to just help to fight off anything. Parts of them will respond to many, many different kinds of pathogens, and they will be triggered by specific responses, but those will be triggered under many different specific responses. Whereas the adaptive immune response is something that we do. We respond to the measles virus, or we're responding to rabies, or we're responding to, a staphylococcus infection. And as we've already covered, it relies on the production of antibodies. And this is also important. It helps us to develop immunity that's long-lasting, and that the second time we see something, we respond to it better. So when we see something for the first time, that's considered the primary response, and subsequent later exposures are going to trigger a secondary response. Just to remind you, because again, I told you the innate and adaptive immune responses, they talk to each other. They're actually integrally linked. So our first line of defense is the skin and mucous membranes. They're secretions in the normal microbes of our body that live on the skin and in the mucous membranes. Our second line of defense, if you penetrate the skin, is white blood cells called phagocytes that will engulf microbes. Our ability to cause an inflammation, bring a lot of cells to the site of injury. Fever, artificially boost our body temperature, and a variety of antimicrobial proteins, including the complement system, including transferrins, including BPD, all kinds of things, interferons and whatnot. So the antimicrobial proteins that we talked about most are the complement system. Where are these complement proteins found? Where are they in the body? They're in the bloodstream, they're also in the lymph, they're also in your tissues. They're everywhere, all the time. Are they normally active? They're not, no. Why? Because they initiate these very dangerous things that we need to protect ourselves, but if they were on all the time and there wasn't anything to attack, they would attack the body, okay? So they're normally inactive. Now, this central cascade I talked about, remember the complement has how many proteins? 9, okay? But this core is initiated by one of them. Which one is it? C3. Exactly. And so if you turn on C3, what are the 3 things that definitely happen? What does it initiate? There are 3 different outcomes of complement activation. Inflammation, opsonization, right? Painting or coating to make something visible, and poking the hole, cytolysis. Mac attack, poking hole, cytolysis. Now, how do we turn this cascade on? What are the 3 ways we can do it? What's the classic pathway? Classic pathway is the first one that was discovered, and it's really linked to the adaptive immune response. Adaptive immunity relies on what? Production of what? Antibodies, right? So if antibodies find their targets, that's one way you can turn the complement system on. So specific response has this generic effect. So that's one way, the classic antibody-antigen pathway. There's two others. The alternative to the classic is triggered by what three proteins that are pattern recognition proteins? However it helps you to remember BPD. I don't know how you're going to do it, but find a way. BPD, which recruits C3 directly and turns it on. And also the third pathway, it's named for that protein that binds to mannose, binds to carbohydrates that are found on bacteria. Lectin, the lectin pathway. So we've got classic, alternative, and lectin are all the ways we can turn on this pathway. Okay, so this is again just to remind you, we've got two lines of defense in innate immunity, your skin, mucous membranes, secretions, and microbes. Second line of defense being white blood cells that can resolve things, inflammation, fever, and various antimicrobial substances. And right now we're going to move into the third line of defense. Okay, this is the adaptive immune response. This is focused on the abilities of two cells, two white blood cells, T cells and B cells, and the antibodies that B cells produce. A little history. So over the years, as people were learning about microbes and what they can do, the understanding of how our adaptive immune response works was developing, but they didn't have a really clear picture until various discoveries had been made. So Pasteur was one of the first people to actually document adaptive immune responses, but he didn't really understand what he was seeing. Okay, so he saw that when he injected chickens with a pathogen that was weakened, that normally would make them very sick, but weakened, they would develop immunity against it. Now, he had some ideas about why that might be that were wrong. This guy, von Behring, was working on antitoxins. He won the Nobel Prize at the turn of the last century. And what he saw was that if you take bacteria that make a substance that will kill animals, this he called an exotoxin. If you take a low dose of the survivors, the few survivors that don't die, if you take a little bit of their blood, that blood is then protective. And he called that the antitoxin. So what do we know now is actually the basis for that protection? What were those surviving animals making in their bloodstream that was protective? They were making antibodies, and if you take those antibodies and give them to another animal and then challenge the animal with the otherwise fatal dose of exotoxin, now they can survive. This is one of many ways that we can use antibodies to protect us. It's not the only way. Paul Ehrlich, who is also famous for a variety of other things, was the one who really documented that antibody protection, the antitoxin, whatever it is very specific. So if you challenge an animal with a certain kind of toxin, those surviving animals, their blood is only protective against that specific toxin. You can't protect animals generally against toxins. So it may be surprising to us now, but the idea of specificity wasn't actually really recognized until his work. And the result of that understanding led eventually to actually identifying the antibodies in blood serum. So there's a couple of components to the adaptive immune response. You've got to detect antigens. You've got to make antibodies. You have to have a way to discriminate between yourself, normal cells, and your foreign cells. And it's not fail-safe, right? Because we know that sometimes we get allergic reactions, which really are our body's responding to something as if it's really dangerous when it's not, right? Is pollen going to kill you? No. But we think it's going to kill you. So we just, send all this. We start inflammation, we do all kinds of crazy things. If you eat a mango, that's not going to kill you unless you're me, in which case it gives you like a weird allergic rash. So there are a variety of ways that this immune response can actually be counterproductive. But normally it's really helpful and keeps us alive. So a key to this is the lymphatic system. Now, we're probably more familiar with the vasculature of the veins and arteries that also have a network through the body to deliver oxygen. The lymph is in vessels all through your body in parallel to the bloodstream, and it has sort of specialized areas called lymph nodes where lots of lymphocytes like to hang out. It's filled with fluid that travels through your body and primarily helps to deliver white blood cells to the whole body. Its other main function is that it brings lymphocytes, white blood cells, into contact with antigens. Now there's two sides to adaptive immunity, and this again is a little bit artificial and it's also historical. The names for this are historical. So I'm going to tell you the history very briefly so that hopefully that will help you to remember it. So there is in your bone marrow both the B and the T cells, the basis of adaptive immunity. And one branch of that is called humoral immunity and one is called cellular immunity. The reason that one branch is called humoral is because hundreds of years ago, when people didn't really understand everything that we know now about the human body, they did understand that body fluids were important and that there were things in body fluids that did stuff. And those body fluids were called humors. So in our fluids, there is, in fact, something very helpful that's important for adaptive immunity. Do you know what it is? It is the key to your adaptive immune response. We've said this word like 10 times already in the last 5 minutes, antibodies. There are antibodies in all your body fluids. And so basically humoral immunity is the antibody part of it. But what cell type makes antibodies? We just discussed this. Those are B cells. So the B cells are considered the half of your adaptive immune response that's humoral because of this historical word, humors. That leaves the T cells, which we just call our cellular immunity. And it is sort of logical because the T cells have a local effect. They're really important, but where the T cell is, it's working and doing whatever its job is. Whereas B cells have both a local and systemic effect because if they make antibodies, then their activity can have a very distant outcome, right? So, in the bone marrow, all of the white blood cells and red blood cells are made. Two of them, the Bs and the T cells, are important for adaptive immunity. And one of these cells stays in the bone marrow and differentiates there. That's the B cell. And one of them is born in the bone marrow, but migrates very soon to the thymus, where it differentiates into a T cell. How can you remember that? They're both born in the bone marrow. But T cells go to the thymus, which starts with T, and B cells stay in the bone marrow, which starts with B. Both of them eventually go into lymph nodes and in other lymphoid tissues like the spleen and migrate through the body in order to try and find their antigens. Okay, so this is in text basically. Both types, T and B cells, develop from stem cells in the bone marrow. Importantly, usually, in order to do anything, they have to have a second opinion. We're going to sort of see what that looks like. There is humoral immunity, which is based on B cells. Again, they mature in the bone marrow. They make antibodies that target things we call antigens on the surface of foreign invaders. And cellular immunity that's due to T cells. T cells mature in the thymus. And they also are looking at antigens, but in a different way. So we're going to clarify those differences. But first, let's talk about B cells and how they make antibodies. So here's a B cell, and it's got an antibody on the surface. And what happens first is that it binds to something that that antibody is specific to. That thing is called the antigen. Very simplistically, and we're going to talk about the way that this is a very simplistic picture, that can trigger this antibody-antigen reaction can trigger the B cell to differentiate into a plasma cell. And this is the cell that makes tons and tons and tons of secreted antibodies that look exactly like the antibodies that were on the surface of the original naive B cell before it was ever looking for its target. These antibodies go and coat their target, and that acts as a flag for your non-specific immune response, your phagocytes, to go and gobble that up and get rid of it. A couple of things that I want to say about this. This B cell made this antibody before it ever found that guy. In your bone marrow, your B cells are made, and every one is a little bit unique. There's a region of the DNA that makes this end of antibodies that in each B cell gets a little bit shuffled. So every B cell is born with a slightly different end on its antibody. This B cell, all of its antibodies are looking for the same thing. So the whole B cell is covered with the same antibody. And as I said, it's born with this desire to bind to that thing without ever having seen it. So it's not like something comes into your body and then the antibody's made specifically tailor-made to that thing. It's just lucky that we have so many different kinds of B cells with so many different specific targets that one of them happens to have the right shape and binds to the antigen. I mean, we have tons of B cells, so, you know, most of the time there's something that's going to bind a foreign invader and recognize it. So this is a naive B cell. Once this antibody finds what it's always been looking for, that's what triggers it to become this specialized cell type. And that allows it to make lots of antibodies. So let's just take a step back and think about what the difference between this naive B cell and this plasma cell might be. What kind of chemical is an antibody? It's a protein, right? It's made from the DNA, and it's made for either to be put on the cell surface or later on to be secreted. And both cell surface proteins and secreted proteins are the ones that when you make them, let's think back, okay? We've got our DNA, we make a messenger RNA, right? Where does it go? Goes to the ribosome. What kind of, in eukaryotic cells, we've got two kinds of ribosomes, right? We've got ones floating around in the cytoplasm. We've got ones that are attached to what? The endoplasmic reticulum, rough ER. Smooth ER is where there's no ribosomes. Rough ER is where there is ribosomes. If a messenger RNA is being read by a cytoplasmic ribosome, that means that protein gets to be kept by the cell. If it's going to a ribosome that's sitting on the ER is a special, basically, production factory that ships things outside. So if you want to make a lot of antibodies for shipping, then you have to turn into the kind of cell that has a crap ton of ER and lots of ribosomes. Okay, so you don't want to be that cell if you're just floating around looking for something, but you don't have to make a lot of antibodies. It would be a waste of time to have this specialized apparatus. You have a little ER because you've got to make some proteins. But once you see that, oh yes, I was right, this thing I was looking for really does exist, I need to make lots of this, you differentiate it into a plasma cell, and the way you look is quite different. We'll see a picture of that in a second. Okay. So how do antibodies actually help us? Once you make antibodies, so the antibodies help you to find something that shouldn't be there. Okay, that's obviously helpful. But why then do you want to make a whole bunch of secreted antibodies? You've done your first job, which is to find out the thing is out there. But now we're making a whole bunch to coat this microbe in order to, A, flag it for destruction, right? Being coated with antibodies is a flag for destruction. But also, if you coat something in antibodies, you prevent whatever's on the surface of this microbe. For instance, what if it's a virus? Remember, viruses have spikes for attachment. You can actually prevent that attachment. So you can prevent it from fulfilling part of its life cycle. So you can antibodies are helpful by directly coating just to prevent the microbe from doing the thing it's going to do next, attaching to whatever cell it wants to attach to. And also it acts as a flag for phagocytes to consume it. So I'm going to come back to this, but this step here actually requires a second opinion. And we're going to talk about that second opinion. It requires a T helper as a second opinion, but we're not going to talk about that yet. I'm going to first talk about T cells. So cellular immunity and T cells. How T cells work is there are several kinds actually, and of the several kinds, I'm going to focus most on and test you on only two: T helper cells and T cytotoxic cells. T helper cells are the ones that are important for giving those B cells a second opinion. T cytotoxic cells are important for checking your body cells to make sure nothing weird is going on. They have similar modes of activity, but we'll talk in more detail in a second about the differences as well. But important to just know that what they do is they inspect other cells, okay? So if they're T helper cells, they're inspecting what we call antigen-presenting cells. I'll define that in a second. But they are inspecting cells that have already encountered some kind of pathogen or some kind of microbe, and they're giving a second opinion to that cell. So the cell has already found something, like the B cell has already found something. Now the T helper cell has to come along, and they have to have a little conversation in order to move forward. So that's why they're called helper cells, because they provide a second opinion. T cytotoxic cells are actually patrolling your body, making sure that your body cells are not infected with viruses, not infected by bacteria. There's nothing weird going on. They also help to protect you against cancer. And when they find a body cell that is not acting right, they kill it. But in order to kill it, they have to have a conversation with the body cell. So there's a little bit of a back and forth there. Before I talk about that in more detail, I want to talk about antigens, because we've been using this term a lot. And so here's the definition. Antigen is a substance that causes the body to produce specific antibodies or to sensitize T cells. So what does that really mean? Most often, antigens are either proteins or sugars. What other kinds of molecules are found on living organisms? We've got protein, sugar, and what else? Lipid. Lipid is the hardest to detect. Lipids can often go unnoticed. So if you're trying to protect yourself from being noticed, you might cover yourself in lipids. It's just because the way that proteins and sugars are, is their structure is easier to see. Antibodies will interact with epitopes, and I'm going to sort of talk about the difference between epitope and antigen. But basically, an antigen is sort of a big thing that can be seen, and an epitope is a little piece of it. That's the specific piece that is seen by one specific antibody. So just to kind of diagram what that looks like, let's suppose that this is a cell, and there's something sticking out of it that can be seen. That's an antigen. But there are different parts of this that might be recognized by different antibodies. And these individual parts of the same antigen are different epitopes. There's another term you have to know. Again, you could be tested on this, and I'm just going to give you a picture in a second to help you remember it. But it's called a hapten that can be antigenic, but it's so small that it would go unnoticed unless a carrier molecule comes around and rounds up a whole bunch of them and makes it easier to see. So here's just again, a nice diagram of the difference between an antigen and an epitope. So antigens are parts of the cell wall that are repeated, okay? So you'll see that this part of the cell wall that is visible is repeated. And they're found on the bacterial cell. It might be some kind of protein that it makes, or might be some kind of sugar combination that it makes that's visible. But there are small sub-pieces of that that are specifically recognized by antibody A binds this part of it, antibody B binds a different part, antibody C could bind the other part. So those specific regions that are bound by this antibody are called epitopes. And you'll also notice that they have these different shapes and that they match the antibodies in different ways. So this antibody's end right here at the end of the two arms of the antibody is specifically looking for triangle things, and this antibody is looking for circular things. Again, haptens are just like really small antigenic particles that have to be gathered up and presented in a group on a carrier molecule so that they don't go unnoticed. We also have to talk about antibodies. So yes, they are proteins. They're called immunoglobulins for immunity, and globulins because they, just the way they fold up. They bind to epitopes, and if we look at a typical bacterial cell, there's lots of epitopes on it. So you can find those, again, it's sugar and protein combinations that can be seen, and there's lots of them. For instance, on the flagella fimbriae pili, the surface, so this is a blow-up of the surface, there could be lots of sugars and proteins sticking out that antibodies can find, or on the cell wall. Now, if you have an antibody, the number of binding sites it has is called its valence, and the minimum number is 2. So all antibodies are at least bivalent, and that's because they have that shape I showed you, that Y shape, right? So literally they look like Ys, and at the end of these two arms is where it binds the thing it's looking for, and this stem is just kind of a structural thing. Each of these arms binds the exact same antigen, and these are made by the same gene. There are 5 classes of antibodies. You may have heard of these. If you've ever looked at profiles of antibodies or done any kind of immunology, you may have heard of these terms. IgG, IgM, IgA, IgE, and IgE. And we're going to talk briefly about all of them because there could be a matching question on a test where you have to know, in brief, what each one of them kind of does, what it looks like, where it's found. So here's just the general structure of your generic antibody. There's 2 pieces encoded by one polypeptide and another one, and then that's repeated. So this is the same gene. It makes a bunch of polypeptides that come together, and then this other gene makes this other part. And it's this end of the 2 ends of the Y, like little hands that are looking for something specific, and this part is what's variable. This is going to be looking for circles or squares or triangles or star shapes or whatever it's looking for. Antibodies are specific to what the antigen they're looking for. Their shape is extremely specific and variable, so their fit is very precise. And it depends on weak reversible bonds. So let's just cast our minds way back to chemistry, chapter 2. I know, that was like 75 million years ago. But remember, we talked about 3 kinds of bonds. We talked about covalent bonds, which are really strong electron-sharing bonds between atoms, which is what's holding the amino acid, all the amino acids in this polypeptide chain together, right, by peptide bonds, and is holding the atoms inside the amino acids together. So that's not what is holding the antibody to its antigen, because that would be too strong of a bond. And we also talked about ionic bonds and what else? Hydrogen bonds. Hydrogen bonds are super useful because they're weak reversible bonds. So they hold DNA strands together so you can pull them apart and replicate the DNA, or pull them apart and transcribe the DNA and then put it back together. And they're also really important for antigen-antibody binding. And some ionic bonds are involved as well. Every antibody has a slightly different specificity right here. And you can change that specificity by changing the amino acids right here, which means that you can change the specificity by changing the DNA sequence. And when B cells are born, they have the ability to make just one kind of antibody, and their DNA sequence has been shuffled for each one that's born. That shuffles that little part of the gene that makes the antibody. So everyone has a different site. Okay, so remember I said valence in this context refers to the number of binding sites that an antibody has. So in this picture, how many antigens can this antibody bind to? Or how many sites are there to bind the antigen that it's looking for? There's two, right? One here and one here. So this is a bivalent antibody, and that's the most common type. But it's not the only kind. So if you just look at a bunch of antibodies under electron microscope, or this is atomic force microscopy, gives you higher resolution, we can see these little Y-shaped antibodies. So I threatened/promised that I would go through all 5 types of antibodies, so here we go. IgGs, and I'm going to give you ways to remember most of them. IgGs have this very generic bivalent Y shape. That's what they look like. This is how I remember them. And we call this bivalent. We also call this a monomer because there's only one antibody, one Y shape. I remember IgGs as G for generic or general because they're by far the majority of serum antibodies. So if you look in your bloodstream, most of them are IgGs. What do? They fix complement. That means they activate the complement system. So if this type of antibody binds what it's looking for, it's going to turn on complement activation. Where do we find them? Bloodstream, lymphatic system, and also in your gut. Why would we want a lot of antibodies in the gut? Because there's a lot of stuff coming in from the outside that's potentially covered in microbes. No matter how carefully you wash your lettuce, I assure you there's still microbes on it. They also can cross the placenta. Why is this important? What would happen if they didn't do this? Your babies would be born with a lot less immunity because they don't have a strong immune system yet. So you give them some of your antibodies initially. So what do these antibodies do besides fixing complement? They enhance phagocytosis. So if they're just floating around, a B cell made them, but now they're floating around on their own. And if they bind to a microbe, then phagocytes will be triggered to see and engulf that microbe. They also can bind to toxins and neutralize them. So again, toxins are usually floating around because some bacteria over here in your hand made them and then it traveled through your bloodstream. So if they bind toxins, they can neutralize the toxins. They can bind viruses. What are they likely doing to inactivate viruses? How do viruses start their life cycle? Yeah, they've got these attachment proteins. So if you cover the attachment proteins or just get in the way of those attachment proteins, we can prevent virus infections. And they're very important, obviously, for protecting the newborn and protecting your fetus. They only live for about 3 weeks. And then they either have to be replaced or they just disappear. So they're important because at birth, that's when your babies begin antibody production. And so initially, infants have IgGs that they got from their mom. And then they start to make their own. So you may know that there's this point where their immunity dips and they're more sensitive than they were when they were first born to infection. Okay, next type we have is IgMs. This is what they look like. How many monomers do we have? Okay, so this is called a pentamer. How many spots for binding antigens? 10. So that means they're actually decavalent, right? Their valence is 10. They can bind 10 things. These antibodies are organized exactly in this configuration. So they're all kind of joined in the center by the stem, and then their hands are reaching out. Why do you think this kind of thing would be helpful? This is going to be really useful at grabbing onto microbes more strongly, right? Because they're looking for something that's probably repeated on the microbe surface. So you maybe could grab the same microbe twice or grab a whole bunch of them and cluster them all together, making it easier for phagocytes to find them. This is how I remember IgMs. M is for multi or mega, so they're the biggest type of arrangement of antibodies. They are about 5 to 10% of the antibodies in the serum. They also, like IgGs, fix the complement. They're also found in the bloodstream, in the lymphatic system, and they're found on the surface of B cells. So they are part of the initial, when a B cell is born and it's making antibodies, this is what's on the surface to try and help them find what they're looking for. And it kind of makes sense you would want something that was extra good at finding stuff.
So they are the weakest without things that
They're the largest collection of monomers. So monomers, or the antibodies can be just as one Y. Sometimes they are found as dimers, you'll see that in a second. And sometimes they're found in pentamers. And this is the biggest one, this arrangement. So IgMs are the biggest form of antibodies. It's not that this part's bigger, it's just that they're arranged together like this. These are actually joined together. So one of the things that they can do, because they're secreted, they're also found in the bloodstream, but they're also found on B cells. So they're great at grabbing things to begin with, and then when you make them and secrete them, they're great at clumping. This is what this means, agglutination. They're great at clumping microbes all together, making it easier. It's kind of like if you're sweeping your floor and all those different dust particles are now in one place, you can get them up more easily. Obviously, the reason that it's helpful is because it has all of these sticking out all over the place. So it's like this sticky tape that's just rolling through and grabbing a whole bunch of stuff at once. It's typically, if you're responding to an infection, this is the first type of antibody you'll produce. Because you want to be aggressive in responding to this, right? So if you're looking at someone's profile of different kinds of antibodies, the levels of this versus other kinds can tell you whether or not you're early or late in your infection. It has a very short half-life, only 5 days. Now, IgAs are the next type. IgAs are dimers, so you'll see these two Y shapes facing out. They're joined together by something called a J chain. And this is how I remember them, primarily for ACHU. This is going to be just a tiny bit confusing because IgEs will come up in a second, but I remember this because what happens when you have a cold? Lots of mucus. Okay, so they're found in mucus. They're also found in other places, but that's one of the, they're about 10 to 15% of the serum antibodies in your bloodstream, but they're primarily found in mucus. They're found in saliva, tears, they're found in breast milk, and they're very high in the body. They provide a lot of protection to your mucous membranes. They basically prevent microbes from attaching to membranes, so they're part of that really early defense against stuff getting into your body through the mucous membranes, which of course we need to have, but they make us vulnerable. Wouldn't it be nice if we just cover ourselves all in skin? Well, that would be weird, but again, they have a fairly short half-life, so you have to make more of them all the time. All right, IgDs are, once again, monomers. I don't have an easy, snappy way to remember them, so you're just going to have to remember them as the kind that we don't have a snappy way to remember them by. No eating in the classroom. They're very rare in the serum. 0.2% of serum antibodies are D-type. They are found in your bloodstream, in your lymph tissue, or lymph fluid, and they're the majority of the ones on B cells. So these are the monomers on B cells. So B cells have these IgDs, and they also have IgMs. Okay, so on the B cell, they're part of initiating that immune response. They're there looking for something to get the whole ball rolling. They have a very short half-life. Last but not least are IgEs. This is how I remember them. They're important for allergic reactions. Look, I have silly ways. You may have your own. I'm just giving you mine. They're super rare in the blood. Mostly where they are is they're found on mast cells on basophils. And those are cells that are important for, cast your mind back to what we just talked about, an innate response. Inflammation. Remember, mast cells are those ones that secrete histamines, right? So they're found on mast cells. They are therefore one of the cell types that's involved in an allergic response. So they are implicated in allergies, but they're also super important in lysing or trying to get rid of a parasitic infection, especially a parasitic worm. Their half-life is only 2 days. Okay, so where do antibodies come from? Who makes them? What cell type? B cells, right? And they're proteins made by the B cell using the DNA of that cell. Are B cells activated all the time? No, they're not. So usually they're naive. They haven't seen what they're looking for yet. Okay, they were born with a specific target, and they just spend their whole life roaming around in your body looking for that and feeling sad. Okay? So, what turns them on? It's a combination of finding what they're looking for, their antigen soulmate, and getting a second opinion from this matchmaker, the T helper cell. How does this work? We're going to talk about some terms, and then I'm going to draw a picture because I think that will be much more helpful. But we have to talk about some terms first, and I'm sorry. So MHC stands for Major Histocompatibility Complex. It's found on mammalian cells. There's 2 types, actually. And one of them is going to be important for activating B cells. B cells are responding to 2 different kinds of antigens, T-dependent antigens. So that would be an antigen that, in order to respond to it, the B cell has to find it and then show it to a T helper cell and get a second opinion. And if the T cell agrees, then it produces a message that says, yes, go ahead, make antibodies. There are some T-independent antigens that directly stimulate the B cell that don't need a second opinion. This is kind of like if your soulmate was just, the chemistry was just so incredible that you wouldn't even bother talking to your mom to see if it was a good idea, okay? No second opinion needed. As I've already said, B cells each only have one specific antibody type. That one type is found 100,000 times on the cell surface. So they have lots and lots of this, just one type, all identical, looking for that one thing that they're specific for. So let's first talk about T-independent antigens. Usually they're giant polysaccharides. So a polysaccharide, remember, is repeating subunits of sugars. And if you have this giant polysaccharide that has whatever you've been looking for, then it's so big that it's going to bind to so many of these antibodies that this is a sufficient signal to turn on the B cell. And the reason that that is the case is that if you see this, it's typically, this is not something you would see in the body normally. So we have this sort of, again, it's kind of a pattern recognition thing, but it's still specific. But binding at many, many points all at once can be a trigger for the B cell. So where do we find this? Bacterial capsules and the lipopolysaccharide layer of gram-negatives, which is nice because these very things are also helping those microbes to escape detection by phagocytes. So, there's a little bit of give and take in these structures of these pathogens. Okay, so what's really happening? This is where I'm going to draw you a picture. So if you are a B cell, then if you're a naive B cell, and you haven't found your antigen yet, but still you have antibodies, all the same kind, in the hopes that you'll find what you've been looking for, all over the place. And then one day, along comes a microbe that has that thing that you've been looking for, and you bind to it. And what happens is, it triggers the B cell to take this in, chop it up, and then put it on the surface in something called the MHC2. So it actually chops this up and puts the pieces of it in here. So what I'm going to ask you to do is think about this, which is a protein complex. It's a bunch of proteins on the surface of the cell. But I wanted you to think of it as a little basket that you put stuff into. So the B cell, if it encounters what it's looking for, chops it up and puts the pieces into this basket, the MHC2 basket. Now, in order to be activated, usually the B cell needs a second opinion. Who needs to provide that second opinion? T helper cells. Okay, so let's make one of those. Here's our T helper cell. Now, what do T helper cells have? They have a couple things. They have something called a T cell receptor. And this T cell receptor has a certain similarity to an antibody in the sense that it's found on the surface. And it is specific for antigens. These are antibodies. These are T cell receptors. The difference is that this T cell receptor is not bivalent. It only has one spot to bind things. It's made independently of the B cell. So just like the B cell, when the B cell is born, this part of the DNA sequence that encodes this end of the antibody is shuffled. The part of the T cell receptor that's binding antigen is also shuffled in T cells. So bunches of T cells are born with very unique, specific T cell receptors. And just like the B cell, those T cells go out into the world looking for that thing and may or may not ever find it. So there's the T cell receptor. A major difference between the T cell receptor and the antibody is that these are only found on the surface. They're never made and secreted for export the way that antibodies are. So that's one thing that's there. There's another thing that's called a CD. That stands for complex of differentiation. And I don't expect you to remember that. I do expect you to remember CD and what I'm about to tell you about CD, but I don't expect you to remember complex of differentiation. Now, have you heard of CD4 and CD8 cells? Does that sound familiar at all? In what context have you heard of this? HIV biology? HIV, the virus, gets into CD4 positive T cells, which is an immune component of your body, right? And actually destroys the ability of those cells to work. So we're about to find out why that is the case. So here's our T helper cell. It has these two things. What are they for? The T cell receptor, well, first I'm going to talk about what the CD does. The CD likes to bind to the MHC2. And it basically sort of grabs onto it, and what happens next is that allows the T cell to use its T cell receptor to look at what's inside this MHC2 basket. And if it finds something that it's looking for, then it's going to activate the B cell by secreting cytokines, and that's going to cause the B cell to differentiate into plasma cells and memory cells. You'll see that in a picture in a second and not in my crazy scrawl. But basically the CD binds to the MHC2 basket, and the TCR, the T cell receptor, looks inside of it. The T helper cells all have CD4. That's what they have on them. So that's why T helper cells are CD4 positive. And that's the kind of interaction they're going to have. So let's have a quick look at that on this picture. Again, here's our B cell, right? Here's its antibody. It's been looking for this antigen. When it binds it, brings it inside, chops it up, and puts it into the MHC2 class II basket. This T helper cell uses the CD complex to bind to the basket and uses its T cell receptor to look in the basket. And if it finds something that it's looking for, it activates the B cell, and that helps to turn it into a plasma cell. It also, this interaction actually activates the T helper cell to make more of that kind of T helper cell so that it can go and activate more B cells. Now, this is a subtle distinction, and one I probably won't test you on, but I do want you to understand it. here's what this antibody's been looking for, okay? It takes it in, it chops it up into smaller pieces that get put in the basket. That's detected by the T cell receptor. So is the T cell receptor looking for exactly the same antigen as the antibody on the B cell? Not necessarily. So it's responding to a smaller piece of that. This antibody is pulling on something that might be a little bit bigger than what it's actually touching directly, and then chopping it into pieces. So the pieces that go in here are not going to necessarily look identical to this. So you don't have to have perfect complementarity, but you do have to have a specific interaction. So what is happening in the T cell when it verifies for the B cell that what it's found is something important? This is another way of looking at it. So here is the antibody binding this epitope of the bigger antigen, right? And this green part is the part that's actually binding, but it's pulling on this whole thing. It takes it inside, it chops it into small pieces, it puts those pieces into these MHC2 baskets, and then T cells use their CD right here to bind to the basket and use their T cell receptor to look in the basket, and depending on what's in there, might trigger the activation of the B cell. I don't want to go into this in excruciating detail. Suffice it to say that activation causes the B cell to differentiate into, a lot of them will differentiate into plasma cells. So it causes a lot of cell division of that particular B cell. So it used to be unique. Now it makes a whole bunch of copies of itself that then differentiate into plasma cells. Some of them are reserved as memory cells, which help to maintain your immunity long term. So again, B cells differentiate into plasma cells, lots of those. These are the ones that have a lot of rough ER, like I told you before. They have these big factories now to make lots and lots and lots of secreted antibodies. And a few of them are kept as memory cells. So we have now extra ones of this particular kind of B cell. And there is something called clonal deletion to get rid of harmful B cells that I won't talk about. Just to give you a sense of what that looks like, we're going to see today. This is what a lymphocyte looks like. So B and T cells both look kind of like this when they're naive. These are what? This is a blood smear. So what are these cells? What else besides white blood cells is in your bloodstream? Red blood cells. We're going to see blood smears today. This is exactly what it's going to look like, and you're going to see lots of red blood cells, the most common cell type, and occasionally you'll see a white blood cell. So this is a lymphocyte. We don't know what kind it is. In order to see what kind it is, we have to look more closely using electron microscopy. Here's what a T cell looks like, and here's what a plasma cell, a differentiated B cell looks like. And again, you can see here's the nucleus, and then look at all of this. It looks like a fingerprint, I know, but all of this is just, this whole cell is filled up with ER, which is pumping out thousands of antibody proteins every second. And that is how we can get a rapid response to something that we need in order to combat some pathogen. I'm going to sort of skip over this stuff. Basically, the antibody titer gets higher and faster in the secondary response next time you see something. You should watch this video. We don't have time to see it in class, but you should watch it. And don't forget that when antibodies bind to their antigens, it activates the complement, right? It causes cytolysis, opsonization. It causes, also it can neutralize things, so prevent viruses from infecting things, prevent bacteria from attaching to things. It can neutralize toxins. So antibodies have a variety of important outcomes. And we're going to talk about T cells next time.